Just like eukaryotic transcription is a lot more complex than prokaryotic transcription, with many general transcription factors such as data binding protein, TF2D, TF2I, in place of a sigma factor in the case of prokaryotic transcription. Eukaryotic gene regulation and eukaryotic transcription factors tend to be more complex than prokaryotic gene regulation. A second difference between eukaryotic gene regulation and prokaryotic gene regulation is that in prokaryotic gene regulation, the cis elements, so the DNA binding sites where transcription factors such as the repressor and the catabolite activated protein, CAP protein, bind are very close to the promoter and the transcription start site. In eukaryotic gene regulation, these binding sites where transcription factors bind can be very far from the promoter, hundreds or thousands or even hundreds of thousands of base pairs away. A good example of how eukaryotic transcription factors act, or some general features of eukaryotic transcription factors, is the regulation of galactose catabolism in yeast. Just like E. coli are a model organism for the study of prokaryotic gene regulation, Baker's yeast, or Saccharomyces cerevisiae, is a model organism for the study of eukaryotic gene regulation. In the galactose or GAL pathway, a set of enzymes are responsible for importing galactose into the yeast cell and then processing it progressively until it's converted into glucose 1-phosphate so that it can undergo glycolysis and then enter the Krebs cycle for harvesting ATP. And there is an enzyme called GAL2, which is responsible for the import of extracellular galactose into the cell, and therefore this would be similar or analogous to permease in the lac operon. Then GAL1 converts the intracellular galactose into galactose 1-phosphate. There's another enzyme called GAL7 that converts galactose 1-phosphate into UDP galactose. GAL10 then converts that into UDP glucose, and GAL7 catalyzes the reaction that converts UDP glucose into glucose 1-phosphate, which can then undergo glycolysis. Now, just like in the lac operon, one expects that when there is no galactose, there will be very little transcription of these GAL genes, GAL7, GAL10, GAL1, all three of which are on chromosome 2 of yeast, and also of GAL2, which is on chromosome 12. However, when their galactose is available, there should be a high level of transcription of these genes so that galactose can then be catabolized and utilized for harvesting ATP. And this is indeed what happens, and there is a 1,000-fold increase in the level of transcription of the GAL pathway genes when galactose is available compared to when there is no galactose. Next, what underlies, or what is the basis of this increase in transcription. It turns out that near these GAL genes are DNA sequences called upstream activating sequences, or UAS. And these DNA sequences are called upstream activating sequences because they lie upstream of the direction of transcription. So for example, GAL7 is being transcribed right to left, and this is downstream, so in the direction of transcription, and this is upstream. And upstream of the direction of transcription, or upstream of the transcription start site, lie these DNA sequences called upstream activating sequences, UAS, that bind a transcription factor, a protein called GAL4. And GAL4 binds these binding sites as a dimer. So this is one polypeptide of GAL4, and on the right is another polypeptide of GAL4, and these two polypeptides, they stick to each other or bind to each other, forming a dimer. And this GAL4 binds to the upstream activating sequences, and one upstream activating sequence also called an enhancer could bind several GAL4 dimers. And what the GAL4 protein does is it recruits RNA polymerase to the GAL7 or all the GAL promoters to initiate transcription at a rapid rate, so that these GAL genes are transcribed when galactose is available to the cell. So GAL4 is analogous to the catabolite activated protein in the lac operon, since catabolite activated protein recruits RNA polymerase to the promoter and increases the rate of transcription of the lac operon. Similarly, GAL4 recruits RNA polymerase to the promoter and increases the rate of transcription of the GAL gene. Now, one interesting property of GAL4, which is also true for most eukaryotic transcription factors, is that the two properties that it has. One is that it has the ability to bind DNA, and the other ability that it has is to recruit RNA polymerase or to activate transcription. These result from different parts or domains of the polypeptide or the protein. And so the part of the protein, the domain that's responsible for binding DNA, is called the DNA binding domain, and the part of the protein that is responsible for recruiting RNA polymerase is known as the activation domain. Now, we know that the activation domain and the DNA binding domain work independently of each other through a type of experiment known as a reporter assay. In a reporter assay, scientists construct a synthetic gene. That means a gene that doesn't otherwise exist in nature. For example, here they have taken a GAL4 DNA binding site and put it next to the LAC-Z gene. Now, if you remember, the LAC-Z gene is from bacteria, from E. coli. And LAC-Z encodes beta-galactosidase, and therefore this is a completely synthetic gene, since part of the DNA sequence of this gene is from yeast, and the other part of the DNA sequence, LAC-Z, is from bacteria. Now, the other property of this synthetic gene is that we can detect when this gene is being transcribed and translated into protein. Now, basically, LAC-Z will make beta-gal, and then one can carry out enzymatic reactions that allow us to detect if there is beta-gal, if the cells are producing beta-gal or not, and how much beta-gal they're producing. And that's why this kind of an experiment is known as a reporter assay, since it reports how much transcription this synthetic gene is undergoing. Now, if we make this synthetic gene, since GAL4 activates transcription, it will recruit RNA polymerase to the promoter, and the gene will be on, it will get transcribed, and we will be able to detect beta-gal. In the second iteration of this experiment, scientists made a mutant version of the GAL4 protein where they deleted the activation domain. So this had only DNA binding. And when they carried out the reporter assay with this synthetic gene, but in a cell that lacked normal GAL4 but had a mutant GAL4 that does not have the activation domain, no beta-gal was detected. So transcription was off. In a third step of this experiment, scientists use the DNA binding domain of a different transcription factor called LEX-A, and once again, just like in the second iteration where the activation domain of GAL4 had been removed, here there was no activation domain of LEX-A, just the DNA binding domain, and therefore no beta-gal was detected. That means the gene was off, there was no transcription occurring. And finally, scientists created with this second reporter gene, which had LEX-A DNA binding site, they performed the reporter assay in a cell that carried a chimeric or hybrid protein which had the GAL4 activation domain, but the GAL4 activation domain was fused to the LEX-A DNA binding domain. And when they did this, they were able to detect beta-gal, which implied that the GAL4 DNA binding domain was able to recruit RNA polymerase to the promoter even when the GAL4 DNA binding domain was not present, and the GAL4 activation domain was fused to the DNA binding domain of a completely different protein. And that tells us that DNA binding is separable. from activation or the recruitment of RNA polymerase. Next, let's consider how the GAL4 activation domain actually recruits RNA polymerase. There are two ways the GAL4 activation domain recruits RNA polymerase. One is by directly interacting with TF2D and Tata binding protein and recruiting them to the promoter, or the GAL4 activation domain interacts with a multi-subunit protein complex called the mediator complex that then recruits RNA polymerase to the promoter. There are other classes of proteins called coactivators, such as C-terminal binding protein or CBP, that are also involved in this interaction between the activation domains of transcription factors and the RNA polymerase 2. Finally, let's discuss how galactose sensing actually works. Now, when the yeast do not have access to galactose, then a protein called GAL80 binds to the GAL4 activation domain and prevents it from recruiting RNA polymerase. So even though the GAL4 protein is bound at the UAS or upstream activating sequences, it is unable to recruit polymerase because GAL80 is bound to the activation domain and interferes with its GAL4's ability to recruit RNA polymerase. Now, when galactose is available to the cell, there is another protein called GAL3, and galactose acts as the allosteric effector. Recall that allostery is when a molecule binds to a protein, causing it to change its activity, and that molecule is called the allosteric effector. In this case, galactose binds GAL3 and causes a conformational change in GAL3, that is, a change in the shape of GAL3 that allows GAL3 to bind to GAL80. And what that causes is that GAL80 is now unable to bind the GAL4 activation domain, which means that GAL4 activation domain can recruit RNA polymerase, resulting in transcription of the target gene GAL1 over here. And therefore, just to recap this process, in the default state, that is, when no galactose is available, even though GAL4 is bound at the upstream activating sequences, it is unable to recruit RNA polymerase because GAL80 binds to the GAL4 activation domain. The introduction of galactose into the cell causes galactose to bind to GAL3, initiating a conformational change, a shape change, an allosteric transition that allows GAL3 to bind GAL80, preventing GAL80 from binding the GAL4 activation domain, therefore relieving the repression and allowing GAL4 activation domain to do its job and to recruit RNA polymerase, resulting in the transcription of the GAL system genes only when galactose is present. Next, let us do an example. Predict and explain the effect of GAL1 on GAL1 transcription in the absence or presence of galactose of the following mutations. And the first one is the deletion of the GAL3 gene. So here is the GAL1 gene with the upstream activating sequences and GAL4 bound to the upstream activating sequences with the activation domain and the DNA binding domain over here, and this is the GAL80 protein that binds to the GAL4 activation domain. And let's do this in two scenarios. So one scenario is when there is no galactose available to the cell, and the other is when you have galactose available in the cell. Now, when there is no galactose in the cell, GAL80 prevents GAL4 activation domain from activating transcription or recruiting polymerase, and therefore the GAL1 gene is off or there is no transcription. When galactose is available in the cell, then galactose will bind GAL3 and this is galactose, and then that will allow GAL3 to bind GAL80, and that prevents GAL80 from binding GAL4, allowing the GAL4 activation domain to recruit RNA polymerase, thereby turning on the GAL1 gene. Now, if we have a mutant strain of yeast where the GAL3 gene has been deleted, when there is no galactose, GAL3 plays no role in regulating the transcription of GAL1 genes, and therefore the gene will still be off. However, when galactose is available, in the absence of any GAL3 protein, galactose will have no effect on the GAL80 protein, since it's GAL3 that binds to GAL80 and prevents it from repressing or inhibiting the recruitment of RNA polymerase by the GAL4 DNA binding domain. And therefore, if GAL3 is deleted, then even in the presence of galactose, transcription at the GAL1 gene will be off. In the second condition that we are asked to predict, the GAL80 gene has been deleted. And so we can construct the same two situations as before. You have no galactose and here you have galactose. And once again, if this were a wild type cell, then GAL80 would prevent recruitment of RNA polymerase. Therefore, transcription will be off at the GAL1 gene, whereas when lactose is available, it will act as the galactose is available, sorry, it will act as the allosteric effector for Gal3 and prevent Gal80 from binding the Gal4 DNA binding domain and allowing the Gal4 activation domain to activate transcription, and the gene will be on. Now, if we have a mutant strain of yeast where the Gal80 gene has been deleted, then we do not have any Gal80 in the cell. And what that means is that the Gal4 activation domain will be able to activate transcription even when there is no galactose in the cell, since there is no Gal80 to prevent or repress or inhibit the recruitment of RNA polymerase by the Gal4 activation domain, and therefore the Gal1 gene will be constitutively on. That is, it will be on whether you have galactose or you don't have galactose.